Hello and welcome to Life at PALS. Today we will go over a biology, IGCSE Biology past paper. As you can see, this is a theory paper, so that's the long paper, the paper with the extended answers. So many people think that is probably the most challenging and difficult paper. That's why we go over this today. This is as well the 2023 specimen paper. We will go over this since the syllabus has changed a little bit this year. So this paper is already up to date for the new and changed IGCSE Biology syllabus. This paper is a CIE paper, Cambridge International Examination, and yeah, let's get started. So the first question on this paper is about villi in the small intestine, and yeah, we'll explore what these villi are and what they are good for. So we have an electro micrograph here, photomicrograph of these villi. And the first question is to state the function of the villi. So the villi are found in the digestive system, in the small intestine, after the stomach, after the duodenum, the first part of the small intestine. Specifically, they're found in the ileum. And this part of the small intestine is mainly for absorption. So that is their job, the absorption of nutrient molecules into the blood. So the villi have a very rich blood supply as well. Capillaries are very close to the surface of the villi, so that nutrients can very quickly and easily pass into them. So let's describe the structure of a villus. So villus has a couple of important structures. So if we would draw a picture of a villus like this, then inside the villus there are blood capillaries, and there is some kind of vessel in the center of each villus as well. So, and blood capillaries come in here too, through a venule, and then split up into a rich network of capillaries, good blood supply one more time, and eventually these capillaries will join back together into a venule. So blood out, blood in. So additionally to that, if we zoom into the epithelial cells, the cells on the surface of the villus, what we can see there is, when we look at the cells on the surface, these cells have folding of their membrane as well, a bit like this. And these structures are called, so these are epithelial cells, and these have microvilli, which are basically membrane folds, nucleus, all of these cells, and so on. Epithelial cells, one more time, are generally cells that form skins inside of our body. So since they're on the surface, they are here on the surface. They're basically the skin of these villi. So inside the center, this blue structure is a so-called lacteal, which is part of the lymphatic system. So it's a lymphatic vessel, no? And then we have capillaries, and that should be enough to score our 3 points here. And so there we can say something like that. At the center there is a lacteal, which is a lymphatic vessel and surrounded by many capillaries. And we can write about the epithelial cells, no? The epithelial cells on the surface have microvilli, no? To increase the surface area, no? Increase surface area. And yeah, so important here is, no, don't need to get into explanations of these structures. It only says describe. So obviously, if we would say describe and explain, then we would have to add a little bit more. The lacteals, explanation in red now. Absorption and transport of lipids. This is where the lipids are absorbed into blood for transport of glucose and amino acids. As well, we could write something like, no, there's a rich blood supply to maintain a steep concentration gradient to increase the rate of absorption of nutrients. Microvilli as well, increase surface area. And this is to increase the rate of absorption by diffusion and active transport. Again, this is not asked here in the question, so only in black is what is asked for. So some additional information here in terms of the explanation. Let's see, maybe that's actually the next question. No, it's not. So blood transports nutrients. State the components of the blood that transport nutrients. It's a bit of a trick question. The way that the question is worded may make us think there's something special, some structures inside the blood, maybe some cells in the blood that transport nutrients. No, it's just the liquid of the blood itself where the nutrients are dissolved in, and that part, that component of the blood is called the blood plasma. Now, the nutrients are dissolved in the blood plasma and transported, dissolved inside of it. The nutrients in the blood can be used to become part of cells. State the name of this process. Now, this is a vocabulary question. You need to know this term. It's called assimilation. which means to become part of, to make part of. Amino acids are used to make proteins. Take two examples of proteins that are found in the blood. There are a couple of, or many different types of proteins in the blood. There are hormones in the blood, so we could mention specific hormones, which are all proteins, like insulin is a protein, glucagon. So two examples of hormones. Another substance would be fibrinogen, which is important for blood clotting. Obviously, hemoglobin. Which is inside red blood cell and binds the oxygen, and is responsible for oxygen transport. And another important one would be antibodies from our immune system, are proteins too. So all of these are possible answers for this question. Explain the effect of cholera bacteria on the digestive system. So if cholera bacteria get into our digestive system, what they do is, so first of all, we can state that the cholera bacteria release toxins, and these toxins cause the secretion of chloride ions from the cells of the intestine into the lumen, the inside of the small intestine. So the cells lose chloride ions. And this decreases, this increases, it's getting confused, it decreases the water potential. Small, actually, small intestine up here. This decreases the water potential in the lumen of the small intestine, and that means water moves into the lumen from the surrounding cells. So the cells of the small intestine lose water into the lumen of the small intestine by osmosis, obviously, by osmosis. We have a high concentration of chloride ions inside the small intestine, which means there's a low water potential. So water moves from high water potential in the cells of the small intestine towards the lower water potential into the lumen of the small intestine. And that results in, so this causes diarrhea. and yeah, severe loss of water from the body and a severe loss of mineral ions like the chloride ions. Insulin is a hormone that regulates the concentration of glucose in the blood. Describe what is meant by the term hormone. So yeah, hormone has a couple of important points in its definition. It is a chemical substance, important because it's a chemical messenger, not an electrical signal like in our nervous system. So a chemical substance produced by an endocrine gland. And it is released into and carried by the blood around the body, and it alters or changes the activity of one or more specific target organs. So the points that we get here are for chemical substance produced in an endocrine gland, carried by the blood, and alters activity of the specific target organ. That are our 3 points. Person A and person B were monitored to see how well they could control their blood glucose concentration. They did not eat or drink anything other than water for 8 hours before the monitoring began. They then drank a glucose solution. Blood samples were taken at 30-minute intervals. The blood samples were tested for glucose concentration. The results are shown. So we have a graph here. We can see that it's actually you see that we have 2 persons and 1 person can regulate the glucose concentration quite well, so it just rises a little bit and then fairly quickly decreases back to normal, or almost doesn't increase at all, and then for the other person it rises very quickly to a high level and then continues to rise more slowly and then slowly starts to drop but doesn't even get back to normal where it was before, after 240 minutes, after 2 hours. Let's actually move the graph over here so we have this as a reference a little bit. And here for now. So calculate the percentage increase in the blood glucose concentration in person A. Actually make this a bit larger. Person A from between 60, which is here, and 90 minutes, which should be this point here. So this is 150. This, what is this? 70. $72, I think. Yeah, say 72. So percent increase, that means we find the difference by how much it increased. So final value minus initial value, and we divide this by our starting value, which is the 100%, and we multiply this by 100. So I don't have a calculator at hand right now, but so according to the mark schema, this should give us 136% increase. Oh, sorry, this is not 150. This is 170. So 170 minus 72. All right. Using the figure of 2.1, compare the response of person A with the response of person B after the ingestion of glucose. So we can see for the first 30 minutes, the response is very, actually, no, this is where the glucose drink was drunken. After 30 minutes, so the first 30 minutes, with a similar response. Almost no change. No, but then for the next 30 minutes, so from 60 to 90, both increase, but a much steeper increase for person A. Then so from 90 to what, 180. Now we can say, and then A continues to increase until a peak at 180 minutes of, now we can refer to the number as well, I'm not sure here, no, somewhere there, 215 or something like this. Only 10 milligram per 100 centimeter cube, no? Whereas B already we're going to say no, then decreases but does not reach the original level. B already decreases after a shallow peak at 90-minute mark. Now we can see for person B, he already starts to decrease at the 90-minute mark, slowly back to normal, whereas A continues to rise until the 180-minute mark. Using figure 2.1, explain the response of person B after 90 minutes. So person B looks very healthy. Now the blood sugar level increased a little bit, and then it started to decrease. So that is related to insulin. And so for person B, the increase of blood glucose level causes the secretion of insulin by the pancreas into the blood. The insulin then causes the liver to absorb glucose from the blood and change it to insoluble glycogen. Glycogen is insoluble glycogen. It's a polysaccharide, so it's many glucose molecules chemically joined together. Because it is a polysaccharide, a macromolecule, so it's a size, it's insoluble, therefore it will not affect the blood sugar level or in general the concentration of dissolved substances in the blood. And that should be enough. Well, the points we probably score here for secretion of insulin, 1 point, where by the pancreas, 2nd point, target organ liver, and change glucose to glycogen is going to be the 4th point. Not 1, 2, 3, 4. Maybe we get 1 point as well for liver absorbing glucose, and then a 5th point, 5th potential point for changing it to glycogen and storing it. Person A had a type 1 diabetes. Outline the treatment of type 1 diabetes. Type 1 diabetes is genetic, which means the body is not able to produce insulin anymore, so therefore treatment is taking insulin via injection or a small pump. Other things would be to, one obviously, for this to do this correctly, to inject the insulin at the correct time, a person with type 1 diabetes needs to monitor the blood glucose level and maybe as well a reduced intake of carbohydrates, because carbohydrates are basically sugar, or will be digested into sugar. Question 3. Figure 3.1 shows a photomicrograph of human blood. So here we see red blood cells, most of the cells are red blood cells, and then we see a couple of white blood cells, and this is a white blood cell, specifically a phagocyte. We know it's a phagocyte because it has this lobe nucleus with not just a round nucleus, but has like this weird shape of a nucleus. This is how we know it's a phagocyte. This is a lymphocyte here because it has a very large nucleus that is not lobed and fills almost the complete cell. So that's how we can distinguish between the two. And yeah, both of them are obviously white blood cells, the phagocyte and the lymphocyte, but they have different jobs in the immune response. So, describe the difference in appearance and the function of the three. Describe difference in appearance and describe the difference in the function. So the three cells, we already mentioned, so we have red blood cell. The red blood cell, in terms of its shape, has a biconcave shape. From the side, that means it's a bit extreme, but not like this. So it's like dented in, has two caves like a lens, biconcave. As well, it has no nucleus to make more space for hemoglobin. And it is smaller than the white blood cells. So this is for shape. Its function is the transport of oxygen. Then we have the phagocyte. So we already mentioned that it has a lobed nucleus and its function is phagocytosis of, for example, pathogens. Phagocytosis is a form of endocytosis where a cell basically eats another cell. So we have a large phagocyte that can use its cell membrane to grow basically over another cell, like a bacteria, for example, and by that it eats it, and then it will eventually kill it by that, and it will excrete the waste products. So we'll form a food vacuole on the inside, and it will basically send digestive enzymes there and kill and digest the pathogen. Then we have a lymphocyte. Structure of the lymphocyte, there's a large round nucleus and almost very little cytoplasm. Its function is to produce antibodies, for example. And form, I can write this as well, and form memory cells. All right, that should be more than enough for this question. Figure 3.2 shows some of the stages of blood clotting. A blood vessel breaks, platelets collect at the break in the blood vessel. After that, the platelets send out messenger signals which attract more platelets, and eventually these signals as well cause fibrinogen, a soluble protein in the blood, to change its configuration to form fibrin, which is an insoluble fiber protein that forms a mesh-like structure and traps the platelets and red blood cells to form a blood clot. State two roles of blood clotting. So the blood clot obviously prevents blood loss so that we don't bleed out, and it as well prevents entry of pathogens into the body. Hemophilia is a sex-linked blood disorder. The blood of people with hemophilia takes longer to clot. Figure 3.3 is a pedigree diagram showing the inheritance of hemophilia. So here we have the key, very important. Squares show males, black squares show males with hemophilia, and circles are females with normal blood clotting. So you can see we have two healthy parents here. These two parents have one son, two sons, and two daughters. Now, important is that so I have 4 children, 2 sons, 2 daughters. This woman and this man are not children of these two parents because they are not connected via lines to the parents. So these two individuals are individuals that are probably married to, so this man is married to one of their daughters and this woman is married to one of their sons. The allele for normal blood clotting time is represented by XH. So X with a capital H. That means it's dominant, and it means as well that the gene is located on the X chromosome. So this is a sex-linked characteristic. The gene is on the X chromosome for blood clotting, and it is not on the Y chromosome. So only the X chromosome has it. Now we know that females have a combination of XX, so females always have 2 copies of the gene, whereas the male sex is produced by a combination of X and a Y chromosome, so men only have 1 copy of the blood clotting gene. Allele for hemophilia for the disorder is the recessive allele, X small h. State the genotypes of the people identified as PQ and R. So genotypes are combinations of alleles, and in this case, since it's a sex-linked characteristic, we need to include the sex chromosomes as well. So PQ and R. So we can write down the gender already in terms of chromosome combinations. We know that P is a woman, so P must be XX, and then, yeah, depending on the letter, let's see what this is going to be. Q is a man, so it must be XY, and R is another man, so it must be XY as well. So the easiest one is probably Q, no, because has hemophilia, that means on the X chromosome there must be a lowercase h. Same, quite easy, should be R, because it's a man, only has 1 X chromosome, so it must be X with a capital H. So the bit more tricky one is P. It's a woman that is healthy, or who is healthy, so she doesn't have hemophilia. However, this still leaves us with 2 possible combinations of alleles. It leaves us with the homozygous dominant combination of both X chromosomes having a dominant allele, but we know that dominant alleles mask the effect of recessive alleles, so in terms of phenotype, these 2 combinations are the same. Both of these individuals have normal blood clotting. There is no visible difference between the 2 individuals with these 2 genotypes. So which one is the correct one? Now, to figure out the correct one, we have to look at the next generation. P has a son that has hemophilia. So one more time, the chromosome combination of Q is X small h, Y. Now the father is obviously X with capital H and Y. So the son has a Y chromosome, which means it gets his Y chromosome always from the father, which means it must get the X with the recessive allele from his mother. That means we can exclude this genotype, the homozygous dominant, so it must be the heterozygous genotype. The mother must have 1 X chromosome with the recessive allele for hemophilia. So that is the genotype of P. The couple S and T, so now we are here, are expecting another child. State the probability that the child will have hemophilia. So here again we have to figure out the genotypes of the parents first. Same thing as in the case for these parents. The male is healthy, so that's pretty straightforward. He must have a capital H and Y, so that's our father. So we can make a Punnett square for this as well to use these ones. So this is going to be capital H, so X capital H Y for the father. And since they do already have a son with hemophilia, this leaves us with the same situation as before. The mother must be heterozygous, so have a dominant and a recessive allele. So we can put that into the Punnett square. No? So one set of alleles from one parent goes on the side, the other set of alleles from the other parent goes on top. And now we just complete the rows and the columns, repeating what's in the starting row and the starting column. So here we have X, big H, X, big H, X, big H, Y, and X, big H, X, small h, and X, small h, Y. The probability that the child will have hemophilia. So we have 4 probabilities. One of these 4 probabilities is a child with hemophilia. So it's 1 to 3 probability, or 25% chance of hemophilia. No, probability itself, actually, let me correct this. This would be the ratio of the probability. The probability would be 1 out of 4. So 1 over 4, which is 0.25. No? So this is the probability. Describe what is meant by the term sex-linked characteristic. No? So we already said this. It means that the gene is located on a sex chromosome, and this is the second point. Sex-linked characteristics are more common in one sex than in the other. We can see there are no women in this pedigree chart, only men that have hemophilia, although it's just a couple of organisms. But yeah, there are sex-linked characteristics that are more common in women as well. This is usually when the disorder, the genetic disorder, is a dominant allele. Yeah, but usually it's recessive ones, so it's the men that are usually the ones that are more affected by sex-linked characteristics. Another example besides hemophilia would be color blindness. And there are a lot more men that have or that are colorblind than there are women that are colorblind. Yeast can respire aerobically and anaerobically. State the balanced chemical equation for aerobic respiration. We know respiration uses glucose. Glucose is C6H12O6. We need oxygen to break it down, specifically 6 oxygens, and it will break down to 6 carbon dioxides plus 6 water molecules. A lot of 6s in here. This is probably best memorized. When yeast respires anaerobically, ethanol is produced. Ethanol is a type of sustainable resource that can be made from a wide range of crop plants. It can be used as a biofuel. Table 4.1 summarizes some information about crop plants that are used to make biofuel. So we get some information about wheat, corn, sugar beet, and so on. The biofuel is produced. Energy yield in gigajoule per hectare. In terms of the field, I guess optimum growth temperature, the optimum annual rainfall range for the crops. The country has a mean temperature of 12 to 24 degrees Celsius. The country has a mean annual rainfall of 1000 millimeters. Suggest and explain which cropland would be the most suitable crop to grow and produce a biofuel in the country. So 12 to 24 degrees Celsius, that means we are somewhere between these three. Average annual rainfall 1000, that is, well, this one is the most suitable. Average means it can be more or it can be a little bit less. These two here are 360 to 1000, so the average is quite probably below 1000 in these regions here. Although temperature wise 12 to 24. So in terms of rainfall, this is best. In terms of temperature, these two are best. So that means we can probably pick one. So it's either wheat or sugar beet. I would probably go for the sugar beet actually, because it has the highest energy yield in gigajoule per hectare as well, which is double than wheat and higher than corn as well. So although the rain might be a little bit too high for wheat, the temperature might be a little bit too low. So therefore it makes most sense to go for the sugar beet. So let's say sugar beet. So we have a temperature range 12 to 24 is appropriate. For an 18 degrees Celsius optimum. Then as well, can be a bit too high for the range, but it's still okay. And now the final reason why we actually picked that is that the energy yield is higher than corn and wheat. Now, which are other options? So the final reason why we chose it is the energy yield, which is just higher per hectare. Describe and explain how a reduced concentration of water vapor in the air would increase the movement of water through crop plants. Okay, so this has to do with transpiration. Transpiration is the I can actually probably write this here. Oh, so yeah, transpiration is the evaporation of water in the leaf, specifically on the mesophys, on the surface of the spongy mesophys cells, and then the diffusion of water vapor out of the leaf through the stomata. Now this is transpiration. First evaporation, then diffusion. Now these two processes that happens in the leaf. So if water vapor in the air decreases, that means the rate of transpiration would increase. Because there's a steeper concentration gradient of water vapor outside, very low. So the rate of diffusion of water vapor out of the leaf through stomata increases This will in return increase the rate of evaporation inside the leaf, and more water is more water moves up the xylem by transpiration pull. Sugarcane is a crop plant that is usually grown from stem cuttings rather than from seeds. Describe the advantage of using cuttings rather than seeds to reproduce crop plants. So this is a form of asexual reproduction, which means Plants from cuttings are genetically identical to the parent plants. And retain the desired traits, whereas sexual reproduction would lead to variation in different traits, and this ensures a consistency of the product, of products with high quality, as well cuttings probably grow to a harvestable size faster. Now that would be another advantage. Figure 5.1 shows the marine iguana, Amblyrhynchus cristatus. Marine iguanas are reptiles. State two features that are used to classify animals as reptiles. Reptiles, different from other organisms, have a dry, scaly skin. If we think about other organisms, amphibians, closely related, have a smooth, moist skin. Birds' skin is covered in feathers. Fishes have scaly skin as well, but live in the water, so moist, not dry. Different scales as well. And the other feature has to do with eggs. It's always good to think about eggs when thinking about vertebrates and how they differ from each other. Since they live on land, at least partially the reptiles, they lay eggs with shells, but their shells are not hard as the shells of birds. So therefore they have eggs with a soft, leathery shell. Two structures that are present in plant cells that are not present in the cells of reptiles. Okay, that should be straightforward. Plant cells have cell walls, and they have chloroplasts, or a large permanent vacuole. Marine iguanas feed on seaweed. Seaweed contains starch. State the names of two parts of the digestive system where starch is digested by enzymes. That is the mouth, by the saliva, which contains salivary amylase. Amylase is the enzyme that breaks down and digests starch into maltose. Maltose is then digested by maltase into glucose. And the other part of the digestive system is the duodenum. This is where the pancreatic juice enters the small intestine, and it contains pancreatic amylase. Again, amylase breaks down starch. It's part of the small intestine. It's probably enough to write small intestine. Explain why the shape of an enzyme is important for digestion. So this is our Pac-Man analogy. This is our enzyme. Our enzyme likes to eat triangles and not squares or circles. So a circle does not fit into its mouth, but a triangle does. So this is why the shape is important. The mouth of our Pac-Man enzyme is called the active site, and the active site has a complementary shape to the substrate, which is this substrate. Now, in biology, the substrate is basically the reactant in the chemical reaction. If the two join together and the substrate binds to the active site of the enzyme, we call this an enzyme substrate complex, and this is when the reaction is catalyzed and the enzyme breaks down the substrate or takes two pieces of substrate and builds them up. Also, sugar important for digestion. Enzymes have an active site, which has a complementary shape to its substrate, which allows the substrate to bind and form an enzyme substrate complex. And that means that therefore enzymes are substrate specific. Now only one enzyme, only one substrate fits that specific enzyme. There are many threats to species, such as the marine iguana. Describe ways endangered plant and animal species can be conserved. This is a 5-point question. I think this is quite generic question. We can write quite a lot of things here. There could be things like breeding in captivity, in zoos or reserves. Now this could even be done by something like by artificial insemination or by IVF. Maybe the male or female live in different locations in the world, but the sperm is transported over to the female in a different country so that they can be bred. And this is to build up the numbers. The numbers of individuals, and they could potentially eventually be released back into the wild. Something else could be education programs to inform people about the situation, or something like not to prevent or reduce pollution, which could negatively affect our species, or prevent restrict hunting, or prevent the introduction of invasive species. Now that invasive species are species that don't belong into a certain area, which are usually carried by humans from one place in the world to a completely different place in the world. And in the worst case, the invasive species get into the wild and they thrive there. They can make use of resources, but usually don't have natural predators or competitors, and then outcompete local species, which may make them even more endangered and reduce their numbers even more. As well, important is like monitoring wild populations and protect them from poaching, and increase the number or size of national parks, reserves, and so on. So many different options to answer this question here. Number 6. Wetlands are important ecosystems. Researchers studied the feeding relationships between the organisms in an area of coastal wetland. 6.1 shows part of the food web that they studied. With algae and phytoplankton at the bottom, then a couple of organisms. Here, oysters, water fleas, pipefish, and so on. And we end up with this spotted sandpiper. Complete table 6.1 by giving the name of one organism from the food web for each row. The producer is at the start of the food web. Again, the arrows show the transfer of energy and matter from one trophic level to the next. So algae and phytoplankton. Secondary consumer. So the algae and phytoplankton are eaten by oysters, water flea, and pipefish. These are primary consumers, which means an example of a secondary consumer is either the blenny, which eats the pipefish. It's a tertiary consumer as well. If we look at the food chain, phytoplankton, water flea, mysid shrimp, and blenny, but going through the pipefish, it's a secondary consumer. So the mysid shrimp could be a secondary or is a secondary consumer as well, or the stone crab. An animal that feeds at 2 trophic levels, we already mentioned that is the blenny. As well, the spotted sandpiper feeds at 2 trophic levels as well. So this one is secondary, or tertiary. Now, if we go that way, secondary; if we go this way, tertiary. The functioning of ecosystems relies on the cycling of nutrients. 6.2 shows part of the nitrogen cycle. Nitrogen gas in the air is changed into nitrate ions, which are changed to amino acids in plants, which are changed to proteins in plants, which are changed to proteins in dead plants, which are changed to ammonium ions. So we have a couple of processes here, A, B, C, D. So let's try to figure out what they are. So proteins in dead animals. Are changed to ammonium by decomposers. So this would be decomposition here. And now we have ammonium ions, and ammonium ions are changed to nitrate ions, and the process is called nitrification. Producing nitrate ions from ammonium ions, nitrification. And the type of organism that does that are nitrifying bacteria. Describe how the nitrate ions used in process C enter the roots of plants. This happens at the root hairs. Absorb ions by active transport from the soil, water then by osmosis, and that happens across the cell membrane via protein pumps or carriers using energy. From respiration. State the name of the structure in plant cells where process D occurs. Sorry. Amino acids are changed to proteins. So which cell structure builds proteins from amino acids? These cell structures are the ribosomes, or something we need to know. State the process that occurs at B. Nitrogen gas in the air changed to nitrate ions. This is what we call nitrogen fixation. And a pyramid of numbers for the wetland ecosystem showed that there were very large numbers of organisms at the base of the pyramid and very few at the top. Explain why. Now this is because energy between trophic levels is always lost to the surrounding. Now this is the 10% rule. Not only about 10% of one of energy on a trophic level is passed to the next this means there is less energy available at the end of a food web compared to the start. Now something else we could write now as well. Energy is lost to the surrounding. We could be more precise here, by heat radiation. That's how most is lost. Yeah, I think that's enough. As well, not all organisms are eaten, so some of them die, and that energy is not transferred to the next trophic level either. And that was the last question on this paper. That was quite a long session. I hope you're still here, and I hope I could help you to study a little bit of biology and prepare for your IGCSE exams in the future. I will go over a few more papers, specimen papers, the multiple choice, and the alternative to practical in future screencasts. So tune in if you want to learn more about the other papers as well. Until then, good luck and have a good day.